Hi guys, this is our lab report 3, effects of drugs on NMJ. I'm David.
I'm Jasmin.
And I'm Heidi.
In this study, we focused on how these three drugs, puree, succinylcholine, and verapamil, affect signal transmission at the frog neuromuscular junction. This is the site where nerve impulses trigger muscle contractions. Puree blocks acetylcholine receptors, leading to muscle relaxation. Succinylcholine binds to those same acetylcholine receptors, but causes prolonged depolarization, leading to paralysis. And verapamil impacts calcium channels, which are crucial for neurotransmitter release. Understanding these effects helps ensure safer medical use of these drugs and deeper insights into their physiological roles.
So, similar to the video that you saw just now, no frogs were harmed in the making of this experiment. The methods we used are going to be based off of the NMJ lab in the BIPN 105 lab manual. And the setup for that is going to be shown on the right. So we use the power lab to record the action potentials from the nerve and the muscle. Drugs were also dripped onto the muscle for at least 5 minutes before recording the results.
So now I'm going to explain how we get a muscle action potential from a nerve action potential. So the first thing that occurs is that on an A alpha motor neuron, an action potential is carried down its axon to the presynaptic terminal. This depolarization activates the PQ-type calcium channels, causing the influx of calcium. Acetylcholine then gets packaged into the vesicles, and calcium triggers vesicle exocytosis. And there's about 300 or so vesicle exocytosis per action potential. I am as well going to mention here the synaptic delay. Synaptic delay occurs due to three key processes. The first key process is the time it takes to activate these PQ channels, calcium going through the channels and interacting with the vesicles of the neurotransmitters and the exocytosis of these vesicles. These series of steps accounts for about 0.3 milliseconds of the synaptic delay, which is the longest step. ACH then diffuses across the synaptic cleft and binds to nicotinic muscle acetylcholine receptors that require 2 ACH to activate. This diffusion across the synaptic delay accounts for about 0.05 milliseconds of the synaptic delay, and it's considered the shortest step and occurs rather quickly due to the short amount of distance the neurotransmitters diffuse across. The binding and the activation of receptors on the muscle membrane takes about 0.15 milliseconds. The three events I just described are involved in the synaptic delay. However, in the lab experiment, the synaptic delay is actually much longer than in reality because we recorded the map rather than EPP directly, the end plate potential. This additional time accounts for the period required for the EPP to reach threshold, initiate an action potential, and propagate along the muscle fiber membrane to the recording electrodes. The nicotinic muscle acetylcholine receptor is a nonspecific mixed cation channel where both sodium and potassium can go through. However, the membrane potential at rest is actually close to the equilibrium potential of the potassium. Due to this, there is a larger force to drive sodium through, causing the influx of sodium. This then generates a postsynaptic depolarization as sodium comes in, also known as an end plate potential. The end plate potential is created where the more ACH you have, the larger amplitude of the end plate potential there is. And the EPP must reach the threshold to generate an actual potential. As the end plate potential gets generated, the EPP is only occurring at the local area right at the end plate. To have an AP, the depolarization has to activate the voltage-gated sodium channels on either side of the end plate. Once the map is activated, it can then spread. At the NMJs, they're typically located at the center of the muscle fibers, and the muscle action potential spread from the middle and outward.
So the first drug that we dripped onto the fog was going to be verapamil, and we noticed that there was no change in the conduction velocity. It remained 71.43 meters per second, and also the synaptic delay uh, stayed consistent at about 2.73 milliseconds. There was also no change in the max map or the max cap, along with no change in the threshold for all of the drugs. And the reason for this is because verapamil is an L-type voltage-gated calcium channel that prevents calcium from entering cells during depolarization. And this leads to the frog being unable to contract its muscles. As Jasmine explained, the synaptic transmission at the neuromuscular junction primarily relies on the PQ type calcium channels, which release acetylcholine from the presynaptic synaptic terminals, and acetylcholine diffuses across the cleft to bind to the nicotinic muscle acetylcholine receptors, which depolarizes the end plate. But verapamil blocks the L-type calcium channels rather than the PQ type channels. And that means that the map amplitude would remain unchanged because verapamil does not have a direct role in changing the amount of acetylcholine binding to the nicotinic muscle acetylcholine receptors. And MAP is directly proportional to the amount of available acetylcholine. And the minimum stimulus needed to overcome the threshold and produce an action potential remained the same across all drugs. This is because the A alpha motor neurons, which produce the action potentials, are found on the sciatic nerve, but verapamil, along with the other drugs, are only applied to the muscle.
So for acetylcholine, we noticed that acetylcholine caused an increase in the synaptic delay, which caused it to rise from 2.12 to 2.48 milliseconds. Although the conduction time remained the same, the maximum map amplitude decreased significantly. Mechanistically, acetylcholine binds to nicotinic acetylcholine receptors and mimics acetylcholine. However, because it resists degradation, it stays bound longer, causing continuous depolarization and preventing the muscle from repolarizing. This prolonged depolarization results in muscle paralysis, and clinically, acetylcholine is used for rapid intubation, but it must be administered carefully due to risks like hyperkalemia. Uh, this is a graph showing the cap and map for effects of acetylcholine. So acetylcholine caused an increase. I'm sorry. Uh, this figure shows how acetylcholine affects the kappa map amplitudes. The thresholds for activating these potentials did not change, indicating that the initial response to the stimulus remained the same. However, the max map amplitude decreased significantly from 2.32 millivolts to 1.11 millivolts after acetylcholine treatment. This is consistent with its mechanism of causing sustained depolarization and preventing repolarization, leading to muscle paralysis. Interestingly enough, the cap amplitude showed an increase, but this is just likely due to unrelated things like the drugs, just unrelated factors not in accordance with the drug, such as temperature fluctuations or electrode placement. This table outlines acetylcholine's impact on the conduction time and synaptic delay. Conduction time remained largely unchanged, indicating that acetylcholine did not affect the nerve signal transmission speed. However, map latency was increased, which subsequently led to an increase in synaptic delay. This is consistent with acetylcholine's action of causing sustained depolarization at the NMJ, delaying the initiation of muscle contraction. These results highlight how acetylcholine prolongs the time required for effective neuromuscular transmission.
The last drug that we applied onto the fog is going to be Curare. And in the figure on the right, it displays the effects of Curare on both the cap and the map amplitude. The threshold remained unchanged, meaning that the initial activation potential was similar for both the control and Curare. However, Curare significantly reduced the maximum map amplitude from 8.01 millivolts to 3.94 millivolts. The cap amplitude representing the nerve action potentials showed no significant change as Curare primarily affects the neuromuscular junction rather than the nerve conduction because the drug was dripped onto the muscle rather than the sciatic nerve. And the reduction in the MAP is due to Curare's role as a competitive antagonist at the nicotinic acetylcholine receptors, which means that it blocks the sodium influx and inhibits muscle contraction. So Curare is similar to acetylcholine, so it can also bind to the nicotinic muscle acetylcholine receptor. And when it does that, it decreases the amount of receptors that acetylcholine can readily bind to. And when Curare binds the receptor and end plate potential is not generated, and that's going to decrease the amount of MAPs. So the table is going to highlight the effects of Curare on conduction time and synaptic delay. The conduction time remained unchanged, indicating that Curare does not impact the nerve's conduction velocity. However, the MAP latency increased, leading to a longer synaptic delay. This is consistent with Curare's mechanism of blocking acetylcholine receptors, which delays the initiation of muscle action potentials. The extended synaptic delay confirms that Curare inhibits effective neurotransmission at the neuromuscular junction. So that requires more time for enough acetylcholine to accumulate to overcome the blockade.
So, as a result, this experiment analyzed the effects of verapamil, acetylcholine, and curare on on neuromuscular transmission, and by observing the muscle action potential and the connection time, as well as the synaptic delay, we are able to observe these effects of these drugs. And by understanding the effects of these different drugs on the NMJ, this knowledge is actually extremely valuable for safely administering these medication in clinical settings and understanding the broader implications in human health.
Right.
And
Thank you so
these are our
much.
resources. Thanks for listening.